Hi students, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and in today's video we are going to revise cell cycle and cell division in less than 20 minutes. Yes, so this video is all about the quick revision of cell cycle and cell division, and so I am going to tell you the important points, the important MCQs as well. So make sure you watch it till the end and do not forget to like, share and subscribe to our channel. So first of all, when we talk about the cell cycle, what is it? It is a sequence of events. We know that the division is very important for a cell, right? So when there is a disturbance in surface to volume ratio, when there is a disturbance in nuclear to cytoplasmic ratio, a cell is going to divide, and that sequence of events, right, by which a cell duplicates its genome. This line is from NCERT, so yes, you have to listen to me very carefully. MCQ can come from this part. So sequence of event by which a cell duplicates its genome, synthesizes other constituents of the cell, and eventually divides into two daughter cells. This is what a cell cycle is. And the duration of cell cycle, it varies from individual to individual. Like in the human cell, it is for 24 hours, and for the yeast it will be 90 minutes. So basically in 24 hours a human cell is going to divide once. This is about the cell cycle. So when we discuss the cell cycle, we have two things, the interphase and the M phase. So when you talk about the interphase, it is also known as the resting state, it is also known as the energetic phase, and it is also known as the invisible phase. So it is the phase when the cell is having very high metabolism. It is the phase where your DNA, it is not condensed, it is present in the form of threads, it is tangled, but cell is preparing itself for the division. So here I will give you my famous or my favorite example. It is just like your NEET examination. Like you have to, for that 3 hours 20 minutes, you have to prepare for 2 years, 3 years, so that you do not make any mistake in that 3 hours 20 minutes, isn't it? So same is the case of interphase. So cell is going to spend more than 95% of its lifespan in the interphase, where it is going to prepare itself for the M phase. So here the content duplication, right, the DNA duplication, right, and even the organelle duplication, everything will take place here. And M phase is the phase where actual division will occur. So it is having karyokinesis followed by cytokinesis. Karyokinesis means nuclear division, cytokinesis means cytoplasmic division. So now when we talk about the interphase, we divide that interphase into three phases: the G1 phase, the S phase and G2 phase. You all can see this diagram as well. So when it is the G1 phase, what is going to happen here? The RNA synthesis, protein synthesis, cytoplasmic growth will be there. So it is the interval in between the mitosis and initiation of DNA duplication. That is what your G1 phase is. That is gap 1 phase, growth 1 phase and post mitotic phase. You can note down these points. So cell is metabolically active, it will continuously grow, and most of the cell organelles duplicate. Then when it is the S phase, it is the actual phase where DNA replication will occur. 2C will become 4C, like your DNA content will get doubled. Okay, what is going to happen here? The DNA content will get doubled, students, but the number of chromosome will remain same. That's the key point. So if initially your chromosome was like this, okay, just before the S phase, so after S phase, because duplication will occur, so it will become like this. That is what you need to keep in your mind. So S phase mein what is going to happen? 2C, that is the initial amount of DNA, and it will become 4C, but the number of chromosome will remain same. So one extra question from NEET 2021, even in the S phase, the centriole duplication will also take place, and histone synthesis will also occur in this particular phase. That's what you have to remember. Now let's move to the G2 phase. Again, the protein synthesis will be there, RNA synthesis will be there, cell growth, cell cytoplasmic growth will continue. So this is G1, S, G2. G2 is also known as pre-mitotic phase, because after it there will be the mitosis. That's why we consider it as the pre-mitotic phase. I hope you remember one line of your NCERT, right, when we discuss that. So how do we consider it? We consider it like this, that M phase is in between two successive interphase. So G1S, G2, right, G1S, G2. So before mitosis you have G2, so it is pre-mitotic phase. After mitosis you have G1, so it is post-mitotic phase. So I hope there is no doubt here. Now let's see, after this what will happen? The cell will go for the M phase. So let me tell you, students, about the checkpoint here. So once here there is one restriction point of start point. So once a cell has crossed this G1 point, it will definitely enter in the synthesis phase, it will definitely complete its cycle. That's what you have to remember. And let's say if it will not get the go ahead signal, if after entering in the G1 phase, if cell is not getting a go ahead signal, then it will enter in a G0 phase. So what's that G0 phase? It's a quiescent stage. Right, it's a cell arrest stage. It's a stage when cell is metabolically very active, but it is not proliferating. It is not increasing its number, but metabolically it is very active. That is G0 phase. Because in our body, we do not have all the cells that are dividing every time, like your heart cells, right? Yeah, like your neurons. Okay, so they will be in a G0 phase. So it can be temporary, it can be permanent, it depends upon the cell. So again I'm repeating, cell is metabolically active, metabolism is going on, but it is just that it is not dividing. That's what you need to keep in your mind. Then come to this part, then M phase. When it comes to the M phase, we know that what do we have? In M phase we have PMAT. What's that PMAT? Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. This is what we have. So let's start with the prophase of the mitosis. So when you people talk about the prophase, the condensation of the chromatin material will be there. What will be there? The condensation of the chromatin material will be there. So as I said, this video is for the quick revision. You will get the PDF in our official Telegram group. Make sure you hit the like button. Make sure you share this video. Make sure you subscribe to our channel. So here you people can see the condensation of chromatin material will be there. The centrioles that are absent in the plant cell but present in animal cell, they will move towards the opposite pole. Both chromatid of the chromosomes, they will be visible. Initiation of the assembly of microtubules will occur here, and at the end of this prophase, your Golgi apparatus, endoplasmic reticulum, nucleolus and the nuclear envelope, this will disappear. After prophase, there is the M phase. So in the M phase, we know that all the chromosomes, they will align themselves in the center, right at the equatorial plate, at the metaphasic plate. So condensation of the chromosome will be completed here. Morphology of the chromosome is studied in metaphase. Spindle fiber attaches with the kinetochore. Always remember, this is the centromere, these are the sister chromatids, right? So always your kinetochore will be attached to the always the spindle fiber will attach to these kinetochore, which are the disc shaped proteins covering the centromere. Important point it is. Then chromosome will lie at equator. Now in the anaphase, what will happen? Splitting of the sister chromatid will there. Chromosome will move towards the opposite pole. Then comes the telophase, right? It is just opposite to the prophase. So ER, Golgi apparatus, nucleolus, nuclear membrane, they will reform. Chromosome will reach their respective poles and they will decondense and lose their individuality. And finally the cytokinesis will be there. So if it is the animal cell, cleavage furrow having actin filaments that will form. If it is a plant cell, there will be the formation of cell plate, right? So that will be formed by the Golgi apparatus. That's what you need to keep in your mind. Now let's move to the next part, significance. You know that to increase the cell number, for healing as well, mitosis is required. For proliferation, mitosis is required. Now let's move to the meiosis. So when we talk about the meiosis, it is nothing but it is the reductional division. So what is the meaning of a reductional division? The meaning of a reductional division is it will reduce the chromosome number to half. So if I take the example of a human cell, 46 chromosomes, after meiosis 1, 23, after meiosis 2, there will be four daughter cells having 23 chromosomes. That's what meiosis is. So it will take place in two rounds, meiosis 1, meiosis 2. So meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. So in between that telophase 1 and prophase 2, you will see a gap, that is the interkinesis. What is it? That is the interkinesis. That is what you need to keep in your mind. Is that clear, students? That's the first point that you need to take care. Clear? Now what's the next point? When it is the prophase 1, so here you will be having 5 substages, LZPDD. What's that? LZPDD. Leptotene, zygotine, pachytene, diplotene, dikinesis. Leptotene, zygotine, pachytene, diplotene, dikinesis. So let's remember what is present in these substages. So prophase 1 is the longest phase of these of all the division. Prophase 1, it is the longest subphase. So just look at it. When it is the leptotene, what will happen? Chromosome become visible under light microscope. The compaction will start, the condensation will start. Now next is zygotene. Next is Zygotine. So zygotine, one thing, when it is the zygotine, you need to remember synapses, pairing of homologous chromosome, right? The homologous chromosome having same size, having same centromere position, having same genes, but alleles can vary. They will come closer, they will be paired. And how come they will be paired? Because the synaptonemal complex will form in between them. So by this time, you are going to call them as bivalent or the tetrad. Bivalent or the tetrad. Now LZP, paquitin is the substage where what is going to happen, where crossing over will be there. Okay, where crossing over will be there. What is going to happen here? The crossing over will be there, students. So this is what you need to remember. These one-liner questions are going to come in your NEET exam, so make sure you take a note of it. So bivalent and tetrad will be very clearly visible in the paquitin, right? Then appearance of recombination nodule will be there. So recombination nodule is the part, it is the point where, you know, the crossing over will occur, the recombination will occur. So crossing over will occur in between the non-sister chromatids of the homologous chromosomes. What's the meaning of that? That we have to consider a pair of homologous chromosomes. Here you guys can see the pair of homologous chromosome. So these two are the sister chromatids, these two are the sister chromatids, but these two are non-sister chromatids. Okay, so non-sister chromatids of the homologous pair of chromosomes, they will exchange their segment, and the enzyme recombinase is going to play the role here. That's what you need to remember, and that is responsible for the variation. So mutation and recombination, that is your crossing over, it is responsible for the variation. So after that, right at the end, recombination will be completed. So where, you know, that recombination has occurred, we are going to see that that is the site of crossing over. Now next, move to the diplotin. So remember, dissolution of synaptonemal complex will occur in the diplotin. What will be there? Dissolution of synaptonemal complex. So homologous chromosomes, they will separate from each other, right? Like these chromosomes, they will separate from each other. If let's say this is the point where crossing over has occurred, so this right, chromosome will try to separate, right? But this point, this X-shaped structure will remain like this, and we call it as chiasmata. Okay, what's that X-shaped structure? That is known as chiasmata. Is that clear? Is that clear? So this is the longest phase of meiosis, which can last for months or years in some vertebrates, like in the humans. Okay, the eggs can stay in this diplotin arrest. If you remember, female ovaries, it start forming the eggs. Okay, it start forming the oocytes. So that oocytes will stay in this diplotin arrest for many years. We call it as dictate arrest. Hope you can recall it, right, bache? So after this, we have the diakinesis. So when it is the diakinesis, last stage of prophase 1, so here terminalization of chiasmata will be there. If chiasmata is present in somewhere in between, it will move to the ends. The terminalization will occur. Chromosomes are fully condensed. Spindle is prepared, bache. Homologous chromosomes will be, they are already paired, they will start moving towards the center, nucleolus will disappear, and nuclear envelope will break down. Then it is the metaphase 1. So in metaphase 1, always remember, when it is the metaphase 1, you will see 2 metaphasic plates. How many metaphasic plates are you going to see? You people are going to see 2 metaphasic plates. 2 metaphasic plates. Clear? This is what you have to remember. So in metaphase 1, when there will be 2 metaphasic plate, because bivalents, your homologous chromosomes are paired. So in anaphase 1, a very important point, kindly listen to me very carefully, right? There is the separation of bivalent. So homologous chromosomes will get separated. There will be the separation of bivalent. It is not the splitting of centromere. That's what you have to remember. And then finally, what do you have? See, anaphase 1, homologous chromosomes, they will be separated. Sister chromatids remain associated. Then telophase 1, nuclear membrane and nucleolus reappear, chromosome decondense, but do not reach to extremely extended state, and cytoplasmic division will start. So that is what you need to keep in your mind, students. So in anaphase 1, what is happening? Sister chromatids will remain together, but it is the bivalent that will get separated, right? So in telophase 1, there is no complete decondensation of genetic material, no, because immediately the telophase 1, where will it enter? The telophase 1, it will enter in the prophase 2. So that time period, that gap here in between is interkinesis. So students, interkinesis will occur, but let me tell you in meiosis, there will be only one round of DNA replication, means that will occur only in S phase, but there will be 2 times. There will be the nuclear division and the cytoplasmic division. That is what you have to remember. So if your concepts are clear, you'll understand it in a better way. Understood? Understood? So after telophase 1, let's move to the prophase 2. Again, nuclear membrane disappear, chromosome again become compact. Then in metaphase, again you will see it is just like mitosis, right? That haploid, that chromosomes, they will arrange here, that half number of chromosomes, they will arrange on the metaphasic plate. Then there will be the splitting of, yes, splitting of centromere will be there when you talk about the anaphase 2. And finally telophase 2, right? So meiosis end with telophase 2, 2 group of chromosomes once again get enclosed by the nuclear envelope. So we have covered all the important points here, all the MCQs here. Even after this session, even if you will pick up your NCERT and you will start practicing the MCQ, you will see that we have covered all the MCQs. So this is all about the meiosis. And if I talk about its significance, so it will conserve specific chromosome number of each species in sexually reproducing organism, and there will be the reduction of chromosome number by half. And then genetic variability will increase, the variation because of the crossing over. So I hope you enjoyed this video. Do let me know in the comment section, students, right? And make sure you subscribe to our channel and let me know which topic you want to revise next. Okay, so take care, revise your concepts quickly. You can even increase the speed of the video, and make sure you quickly recapitulate all the concepts like this, because I really want my students to score 360 out of 360 in biology. My minimum score is 330. I want my students to score above 330. So you let me know, you please tell me in the comment section that what is your minimum marks for the biology. So stay tuned, take care. Thank you so much, people.